All right, I think we will make a start. Hello, everyone. So thank you for joining into this webinar on bioinformatics analysis of DNA methylation data. So my name is Damien Calais. I will be the moderator or the main moderator with my colleague Jerome Croonen of this webinar. So first of all, I would like to make a few comments. So if you have any technical issues like sound problems, please can you use the chat box to let us know if you have any technical problems. Secondly, please use exclusively the QA box to ask your questions related to the content of the webinar presentation. So at the end of the presentation, we will select a few questions from this box and it will be answered by our speaker. And so now let me introduce to you our speaker, Dr. Saul Schwartzman. So Dr. Schwartzman, she obtained a PhD in predictive microbiology and also a theoretical master in bioinformatics with computational biology. She then carried out a 4-year postdoc working on epigenetic mechanisms of metal hyperaccumulation in plants before joining the epigenomic service of Diagenode as a bioinformatician 3 years ago. So that's it from me. So Sol, it's up to you.
Thank you, Damien, for your introduction. You can hear me? Just a sound check.
Yes, very good.
Yes, good. So thank you very much. Yes, so today I'm going to share with you how to analyze data for DNA methylation experiments. And so this webinar is going to walk you through the steps to go from sequencing data to results. So in order to understand better how to deal with DNA methylation from an analytical standpoint, I'm going to start by explaining a little bit what DNA methylation is, how we can study experimentally DNA methylation, and how to prepare the sequencing files in order to extract methylation data from them. And then finally, at the end, I'm going to go through a little bit what the applications for DNA methylation are. So what is DNA methylation is the question. So DNA methylation is an epigenetic mechanism that occurs by the addition of a methyl group to DNA. So a methyl group being CH3. It is present in DNA virus, in prokaryotes and in eukaryotes. And epigenetics is often referred to as an additional layer of information that goes on top of DNA, and it does not affect the DNA sequence. So although virtually all cells in an organism contain the same genetic information, not all genes are expressed simultaneously by all cell types, and that's where DNA methylation is going to play a role. So DNA methylation can occur at different genome locations, such as gene bodies, where usually it causes activation of the gene, promoter regions, where it usually causes an inactivation of the genes, and it can also happen at non-coding areas such as repetitive regions, small RNAs, or pericentrometric regions. And the location of the methylation will often have an impact on the function of the genes, and it will affect the gene regulation. The most widely characterized DNA methylation process is the covalent addition of the methyl group at the 5th carbon of the cytosine nucleotide. And this results in 5-methylcytosine. Also, can also be known called the 5th base of DNA. And although we are going to be focusing in this webinar on 5-methylcytosine, just to remind everyone that methylation can also occur in other bases such as adenines. In human DNA, 5-methylcytosine is found in approximately 1 to 4% of the genomic DNA. In somatic cells, 5-methylcytosine occurs almost exclusively in around 80% in the context of paired symmetrical methylation of a CpG site. So this is when a cytosine nucleotide is located next to a guanidine nucleotide, as we can see here. An exception to this is seen in embryonic stem cells, where a substantial amount of 5-methylcytosine is also observed in non-CpG contexts. So just to explain, a CpG context, as we said, is when a cytosine is close to a guanidine, and this happens both in mammals and in plants. Then we have other two contexts that we talk about, such as CHG and CHH. So this is when a cytosine is close to another nucleotide So H being a nucleotide that is not a cytosine, so it can be close to adenine, thymine, or a guanidine. And then the other context is the CHH, so we have 2 examples here. And as I said, these contexts are not very abundant in mammals, except in embryonic stem cells. However, the 3 contexts are present in plants, and the 3 contexts have biological functions in plants. So we can also talk about CpG methylation based on the density of the CpG. So we can find regions or clusters of CpG, which we're going to call CpG islands. These areas are usually around promoter regions. And then we have around the CpG islands, we're going to have what is called CpG shores, which are the immediate up and downstream regions of the CpG island. So they are usually 2 kbs up and downstream. And then 2 kb up and downstream of the shores, we're going to find the shelves. And anything that is outside these CpG islands, shelves, or shores, we call open sea. And then we might wonder, what is the biological function of DNA methylation? So as I said before, depending on where the methylation is found in the genome, the function is going to be different. So for example, DNA methylation on the gene body is associated with a higher level of gene expression in dividing cells, but in slowly dividing or in non-dividing cells, such as the brain cells, gene body methylation is not associated with increased gene expression. So it is also going to depend on which tissue we are studying. The methylation, for example, on CpG islands results in stable silencing of gene expression. It can also impair transcription factors to bind, and it can also favor the recruitment of repressive methyl binding proteins. And also we can find that methylation in intergenic regions has a role. So for example, when we find methylation in these repetitive regions, we know that there is going to be a silencing of transposable and viral elements. So these elements, if they're expressed, they are potentially harmful, as their replication and the insertion can lead to gene disruption and DNA mutation. So DNA methylation is also going to regulate that. Okay, so that's a little bit of theoretical background. Now, how can we study DNA methylation in the lab, right? So I will discuss technologies that are compatible with Illumina sequencing, and this can be grouped mainly in 2 types of classes. So the first one is based on the use of specific antibodies that are going to target methylated cytosines. And then we use, you can use an immunoprecipitation to enrich your sample on methylated fragments. And the other technology is based on the chemical or enzymatic conversion of unmethylated cytosines into another base. And in this case, because we are going into single base resolution, we're going to have more precise information on which cytosines in the genome are methylated and which are not. So I'm going to start off by explaining the capture-based method, but the bioinformatic analysis that I'm going to explain later on is based more on single nucleotide resolution, so on this type of technology. So on the region resolution level, I'm going to give the example of MEDIP-seq, because MEDIP-seq is a technology that we propose here at Diagenode in the epigenomic services team, and we also sell the kits, so it is available from Diagenode. So MEDIP-seq stands for methylation DNA immunoprecipitation sequencing. This is a technology by which specific antibodies against 5-methylcytosine are used to capture methylated DNA regions. So here we have DNA with the methyl group, and so we have antibodies that would recognize this region and they would bind to it. And then by means of immunoprecipitation, we would just capture all those fragments that contain methylation. And then we determine by sequencing the methylation states of the different regions. So here we can see that when we sequence and we map the reads to the reference genome, we are capable of discovering regions with enrichment, which would tell us where in the genome there was some methylation. These regions are usually of a resolution of 100 to 500 base pairs. It covers the whole genome, and it therefore has no bias towards any certain region, and it's applicable to animals and to plants. The only downside, I would say, is that you can see which regions in the genome are methylated, but you're not going to have information of which cytosines within that region were methylated, or even you don't have a more fine view of what's the percentage of methylation in those sites. So to contrast that, we have the second type of methods that are single base resolution. And so I also talk about these technologies in this slide because these are the ones, again, that we propose as a service. And what characterizes them is that they go from, they differ in how much portion of the genome you can study with this technology. So we start with the whole genome bisulfite sequencing, which is going to have full coverage of the genome. It's going to, for example, in human samples, it can cover up to 20 million CpGs, but it can be applied to any species. And it's I would say it's the most popular approach to use for biomarker discovery, because you have a vision of all the CpGs in the genome. But we have another version, which is a reduced representation bisulfite sequencing. This technology is characterized by the fact that we use a restriction enzyme that is going to enrich for CpG-rich regions, which means we're going to only focus on those regions where there is a lot of CpGs. Therefore, we don't go for the full genome. In this way, we reduce, so instead of getting 20 million CpGs, we can get 4 million CpGs with this type of technology for human samples. For other species, the number of CpGs changes. It is also used widely for as a biomarker discovery tool. And then we also have bisulfite amplicon sequencing, which is it's basically a technology that is going to work targeting on certain regions of the genome. If you have certain genes that you're interested in, then you don't need to go for a whole genome or for RRBS. You can just focus on those genes, and we would just be getting those regions and studying the methylation levels there. So again, it can be applied to any species, and it is usually used more as a validation tool. So if you have already done WGBS or RRBS and you have located some signatures that you want to validate, well, you can do it with bisulfite amplicon sequencing. Just very, very briefly, I'm going to talk about EPIC Infineon bit chip methylation arrays. So these are precasted chips from Illumina that contain already a set of 850,000 CpGs. These were selected based on different requests. So they are mostly genes that are involved in cancer processes. This technology is only applicable to human samples. And the output, because this comes from an array, so the output is different to what we get from sequencing, and therefore it requires that specialized software is used in order to analyze the data. So the bioinformatic analysis and DNA methylation patterns requires a basic understanding of the bisulfite sequencing technology. So I'm going to explain a little bit more here. I'm going to base this bioinformatic webinar on RRBS. Okay, so this, as I said, is reduced representation bisulfite sequencing. Just to explain a little bit more the technology, the first step is using a restriction enzyme called MSP1. This restriction enzyme is going to recognize and target sites that contain this sequence, so CCGG, and it's going to cut between the two cytosines in this way, in a way that that means that all the reads that we keep for the library, they all start by CGG. So it is a technology that is tailored to target CpG-rich regions, which means that it's going to cover mostly promoter and gene body regions. It requires of a bisulfite conversion, and it gives single nucleotide resolution. So I'm going to explain a little bit the bisulfite conversion step, because this is crucial to understand the analysis later on. So as you can see here, we have a strand of DNA where there is a cytosine with a methylated group, and we have a cytosine with no methylation. So basically what happens with the sodium bisulfite is that there is a deamination of the unmethylated cytosine into uracil. So basically the methylated cytosines remain protected and they don't change after the bisulfite conversion. But the unmethylated cytosines, they change, they become uracil. And after there is a PCR amplification in which we use a special enzyme that allows the amplification of uracil into thymine. And so that means that the resulting strand after the bisulfite conversion is going to show cytosines where there was methylation and thymines where there was no methylation. So I'm going to jump straight into the RRBS analysis. The protocol described here is tailored to Illumina sequencing data. Other bisulfite sequencing data can be used, as I said, like WGBS or BSAS. It's also applicable. The only condition is that the starting files are FastQ files. It allows to work both with single-end or paired-end sequencing, and there are no specific requirements in terms of insert size or read length. So what I call the standard analysis here is the process by which you transform your FastQ files with reads into data, or data formatted in a way that you can use for your statistical analysis later on. So there are a series of steps that you need to go through in order to extract from the reads the CpGs that you're interested in. So I'm going to explain a quality control step for the sequencing, then a trimming or filtering of the reads, so to get rid of all the reads that are not of good quality and that are going to mess up your alignment. I'm going to explain a little bit what the genome preparation means that you require to do if you're working with a bisulfite converted DNA. I'm then going to explain the alignment step, the methylation call, and the methylation extraction step. And I wanted to share with you the tools that we currently use. So we use FastQC, TrimGalore, and Bismark mainly for this part. These are all tools that have been developed by the Barbara Ham Institute. So you can find in this website the software if you want to download it. And just one thing to let you know is that this software is going to require that you have installed other tools such as Biotype2, SAM tools, Picard tools, or CutAdapt. And so if you're just starting with DNA methylation analysis, a recommendation would be if you want to install BioLinux. BioLinux already has a set of bioinformatics tools installed. I'm pretty sure all these come there, and so that can get you started. So I'm going to start now by looking at what happens first, right? So what happens first is that you sequence your library, and you obtain from the sequencing facility FastQ files. So I'm going to, the example I'm going to show is based on the NovaSeq sequencing. It's for a for a library that was sequenced in paired end and for 50 base pairs long each read. We can do this analysis with directional or non-directional libraries, but I'm going to talk about directional because our RRBS kit does directional libraries, which means that it constructs the libraries from the original bottom and top strands of the DNA. Okay, and so when we receive the data from the sequencing, we get a FastQ file. So what is a FastQ file? A FastQ file is just a text file. It contains all the reads, all the information for the reads that were sequenced. And for each single read, it's going to have 4 lines. So say if you are sequencing 30 million reads, you're going to have for that sample a file that contains 30 million times 4 lines. So the first line has a read name. If you're working with paired end data, you're going to have a file for your read 1, which is going to be included in the read name. And if you have, and then for your read 2, you would have another file that contains in the name R2. The next line, the second line, represents the read that was sequenced. The third line is it has no information, it's just a plus sign. And then the fourth line is a code that represents the Phred quality score. So what is the Phred quality score? Well, this score is given by the instrument, the sequencing instrument, for each single base that it calls. And it's going to give a measurement of what is the certainty that the base was called correctly. So the Phred scores can go from 10 to 50 as an example. So 10 means that there is a probability of 1 in 10 that the base was called incorrectly, or what's the same, there is a 90% accuracy that the base call is correct. So as you may have already thought, what we want is larger numbers. So we want the bases to have been called with a lot of certainty. So we want the Phred score to be close to 40. So the sequencing instrument gives you these files with this coding here. As you can see in this box below, I'm showing you the correspondence between the symbols, characters, and the Phred scores itself. So the software needs to know which coding of Phred score you're using, because depending on the instrument, the Phred score could be +33 or +64. So you have to know beforehand which instrument you use so that you can tell the software that you're using a certain coding, because it has to be able to decipher this, this line here. So why is that important? I'm going to show you next. When we do the quality control of the sequencing files, we're going to be looking, we use this software called FastQC. And it's going to give you several plots, but I'm going to show here just 3 that I consider the most important ones for checking the quality of the sequencing. So this is the first plot called per base sequence quality. And I'm going to explain. So in the x-axis, we can see all the positions of the read. So as I said before, we were sequencing 50 base pairs. So we have here positions 1 to 50. And on the y-axis, we can see the Phred score. So here we monitor that the sequencing run was of good quality. Basically, we want, as we are seeing here, this good example, we want the line to be all across 36. What FastQC does is it takes all the reads in the file and it computes for every position the median of the Phred score. And then it plots it like this. So here is an example of a bad sequencing run where you can see that as the sequencing advanced, the quality dropped. And there is a lot of variability. So in your next step, when you filter out reads, you will probably lose a lot of them, because if you don't want really to keep reads that have a Phred score below 20. And then you might want to, if you have a lot of bad reads at the end, you might want to trim the reads. So you might lose a lot of sequencing data. So you're basically throwing your money away. And just to let you know as well that this plot comes from one of our projects. This one, I got it from the net. And just to let you know that we're very conscious about good quality sequencing, good sequencing quality, and so we make sure when we sequence here at Diagenode that we get very good quality. The next type of plot that we want to look at is the per base sequence content. This also comes from the FastQC analysis. So here, what we look at, for example, for an RRBS experiment, one of the quality controls you can do is to make sure that your reads are starting as we want them to start, because as I explained earlier, in RRBS, we are excising the fragments of DNA at a certain position. So we know that they will all start with CGG. Therefore, we can monitor that with this type of graph. So we can see here in the first position, this type of graph is like the previous one, where we have on the x-axis the position in the read, and in the y-axis we have here the percentage of bases that we observe for each position. So we can see here that the first position has 45 and 55% cytosines and thymines. And this, so the first position was a cytosine. So since this one can be methylated, we are expecting either Cs or Ts to be there. So this is normal. And okay, sorry. And in the second and third base, we expect to see guanidines, and that's what we can see here indeed, that we are getting the beginning of the read is the same. And so that means that the sequencing is okay, that these reads look like RRBS libraries indeed. And just to show you an example of a WGBS plot, what it would look like. It's different, but typical WGBS plot looks like this. And we have cytosine that drops nearly to 0%, and then the thymidines should be at around 50%, and then we have the adenosines and the guanidines at around 25%. And this was also to show you that depending on the library type, the profile is going to look different. So if, for example, for RNA sequence libraries, the profile is also completely different. So this is expected. Another thing that we check is the adapter content. So this happens if the fragments that we selected during the library preparation are too short. In this case, we see that the sequencer will start reading, and then if the the fragment was too short, it will continue reading through the adapter. So FastQC is also going to detect, is going to check if there is adapter sequence. In this case, we can see that after position 17, a little bit less than 10% of the reads contain an adapter contamination here. So this is something you want to take care of in order to filter the reads appropriately. So the next step is filtering the reads. So we see that we have just checked with the FastQC how the reads look like, and then you want to take into consideration what you've seen in order to clean them up a little bit. So for example, removing the adapter contamination, removing the sequences that become too short after you've trimmed them, or you want maybe to remove an artifact from sequencing. If you have the last base is always an adenine, for example, you would want to remove it. And so we use TrimGalore, which is a software that deals with MSP1 digested libraries. And it can also deal with paired end reads, which is really useful and handy in this type of analysis. So the next step is to prepare the genome, right? So we said that that's one of the required steps here for bisulfite converted DNA. Why? Because, well, as I showed you, when we convert the DNA with sodium bisulfite, the sequence changes. So we need to make sure that the reference genome we're using is adapted. So what Bismark does is like a preparation of the genome. It only has to be done once. So all the alignments that you do afterwards don't require this. You just need to prepare the reference once, and it's stored, and it's used by the aligner. And the preparation, it's like an in silico conversion, where there is a version that is a C to T conversion, and then there is a G to A conversion. During the alignment, Bismark is going to use Biotype-2, which is an aligner, very popular. We use the standard, the default seed length of 20 base pairs with 0 mismatches. And one of the advantages I think it has is that you can parallelize the jobs. So basically what it means is Bismark is going to use Bioty in 2 branches. It's going to align in parallel your files. But with Bismark, at this stage, you can assign more CPUs if you have, and it's going to parallelize more the tasks, which means that the alignment can go a lot faster. So basically with the alignment step, what we're doing is that we want to see where our reads that we have already filtered and trimmed and cleaned, we want to see where they go, right? Where do they map in the genome? So the next step is going to be the alignment of these reads into the genome. And from the alignment, we're going to obtain a file that is going to tell us for each read where it is mapping. So in this case, it would tell us it's in chromosome 1 from 500 to position 500 to 600, for example. So, okay, we have that information. Also wanted to show you when we map the reads for RRBS libraries, how they look like. So we have here an image of a genome browser. You can see underneath there are all the genes. We have here GAPDH, and you can see all these gray bars are the reads that are mapping into this line, which is a reference genome. So you can see very clearly that for RRBS libraries, the reads map in CPG islands, and so that's why it's called reduced, because you're not mapping everywhere, but you're just mapping to certain regions. And if we zoom in this slide, we see that, for example, if we focus, we have here a better vision of the nucleotide. So if we focus on this cytosine, we see that where these are all the reads that mapped in this region, and when there is a SNP, like for example here a T, it's going to be highlighted. So these all T's belong to unmethylated cytosines. And when the read remains gray, it's because there is no SNP. So that means there were cytosines in every one of these reads, which means these were reads where the cytosine was methylated. So just by looking at this, we can see that around 80% of this cytosine is unmethylated, which means that around 20% is methylated. And we can also see that in this bar, where we can see in blue the methylation and in red the unmethylation. So, okay, we can see that visually here, but how can we put that in a format, in a file where we have that information? This, you know, we want the cytosine with the position and with this information of 20% methylation. So here comes the next step, which is the methylation calling. The methylation calling is going to identify every cytosine in the BAM files. So BAM files are like, you can imagine it's like an Excel file. It has a few columns. In each column, there is a different type of information. And then each row contains the reads. And so we can find, it's called BAM. It stands for binary alignment map, binary alignment map. And it's a binary file. You can open it with a tool called SumTools. And when you open it, you have all this information. So for example, column 1 has the read name. Column 2 tells you in which strand it mapped. Column 3 is the chromosome. 4 and 5 are the start and the end of where the read maps. And then we jump to the column 7, where we can see the actual read. And I highlighted in colors all the cytosines. And then column 8 is going to give us this, what we call this methylation call information. So it's going to use a code that is going to tell us for each cytosine, whether if it belongs to a CpG context, so that's the Z's, if it belongs to a CHG context, which is a X, or if it belongs to a CHH context, which are the H's. And also if it's lowercase, it means that the cytosine is unmethylated. And if it's uppercase, it means that the cytosine is methylated. So now we have that information, and what we need to do next, it's called the methylation extraction. With this step, what we do is that we take all that information that we had in the BAM file and we put it in bed files. So files that look like this, where the information is already a lot cleaner. And we're going to obtain 12 individual output files per sample. So it's going to give information on the original top strand, the original bottom strand, and the complementary to those. And it's going to produce these 4 files for every single context. So for CpG, CHG, and CHH. And at the end of this step, what we obtain is a coverage file, which means that we have the chromosome, the start, the end position of the CpG that we are looking at, and then we get finally the methylation percentage. That's what we need to do the more downstream analysis. And this is the end of the standard analysis, basically. So now I'm going to show you quickly an example of a differential analysis. So now that we've created those files, we're going to use them to do comparative analysis. So every experiment, we can imagine most of them, or most of the ones that we see, they always want to compare groups of samples, either if you have control treatment samples or diseased healthy or diet A, diet B. The examples that you can think of, there's always going to be a comparison. So we use a software, a tool called Methylkit. This is an R package, so you can download it from Bioconductor. Methylkit is going to allow us to do another type of QC. So I'm going to show you here the QC, what this QC consists of. It's going to do an analysis of all the CpGs that have been selected, and it's going to plot. So we have this first plot called the percentage of CpG methylation. So it's going to show on the x-axis the percentage of methylation, and on the y-axis the frequency. So this is a bimodal graph, and this is what you want. Actually, you want to see that your samples had a high amount of reads with no methylation and a relatively high amount of reads with full methylation. And then you would expect that in the middle, you have intermediary methylation levels. If you were getting a bump like this, that would mean that probably your bisulfite conversion did not work properly, or that there is another problem with the library or with the filtering of the reads. You would need to check that. But so this is a quality control. And the other quality control is this histogram of CpG coverage. So coverage means how many reads map to a certain position. So this is represented in log 10 scale. So here we have 10 reads, 100 reads, 1,000 reads. So what we're seeing here is that a large amount of reads had coverage a coverage of 10 reads mapping to that cytosine. This is completely normal and expected. So the average for RRBS libraries is around 15 reads. So what's called 15x per cytosine. And then you would see that there's a drop in number of reads as that have more coverage. So this is normal. And what you want to watch out in this type of plots is if you have a second bump here, if you have two peaks, that would mean that there might be reads that have PCR duplication, and you want to get rid of those. So as I said, Methylkit is going to be used mainly for comparative analysis. And before you do the analysis, there's still another filtering that you have to do to your CpGs. So first you want to filter out CpGs that have low coverage. Why? Because you need the data to be reliable. And if you only have 6 reads, well, to say that there is 50% methylation, it's borderline, right? So it doesn't have a lot of, it doesn't contribute a lot. But if you have 100 reads and you have 50 and 50 methylated and unmethylated, well, then that's more reliable. So you want to filter out any CpG that has low coverage. This is a customizable parameter. So you can say what's the number of reads that you want to filter for. And you can also decide the percentage of the samples that you want that criteria to be met. You can say, for example, I want to filter out CpGs that have less than 5x coverage in 75% of the samples, for example. And you can play with those parameters. Another thing you want to filter out are high coverage reads because they might indicate that those are possible PCR duplicates. So you can remove them also at this state of the analysis. And then what Methylkit is going to do is going to normalize all the CpGs for read coverage distribution between the samples. The next step is you perform your pairwise comparison. So Methylkit is going to use a logistic regression to compute the p-values, and the null hypothesis being that there are no differences in methylation between the two groups. So once you have your p-values, then it's going to use a sliding window model to correct the p-values to q-values. And then here you have, again, another step where you can customize the parameters that you want in order to select the differentially methylated CpGs or the differentially methylated regions. So we use a Q-value cutoff of 0.01, and we use a standard methylation difference higher than 25%, but this can be modified. As I said, we've seen, for example, in cancer studies that you can only start to see differences at a lot lower than 25%. So here I'm just showing well, that you can either study differentially methylated CpGs, or you can study as well differentially methylated regions. And when we talk about regions, we would be moving a window at a step size. So both the window and the step size are 1,000 base pairs, so 1 kb, to compute the average methylation of all the samples for those 1,000 base pairs. So just to show you here an example that, you know, when we detect the differentially methylated CpG between two groups of samples, it looks like this, but you can also obtain that type of differences, as you can see here, for regions. If you use Methylkit, you're going to have as a result as well, you can get these pie charts where it's going to show you the percentage of hypo and hypermethylated CpGs for a comparison. If you do the analysis with us, we also provide you with a volcano plot. that is a better way to visualize your CPGs, your differentially methylated CPGs. And we show here the x-axis, the percentage of differential methylation, and on the y-axis, the Q value. So when you look at this type of plots, you want to focus maybe on the CPGs that are on the top right and the top left parts of the plot, because these have higher differences and they are highly significant. Another type of plot that Methylkit outputs is the CPG methylation percentage per chromosome. So it's going to show you, depending on the Q value and on the percentage methylation difference that you use, it's going to show different results. So that's another thing that you might want to do, is to play with those parameters and see how that affects the hypo and hypermethylation in your comparison. And then when you do the analysis with us, we provide you with a histogram that picks the top 100 or the top 20 most differentially methylated genes. For example, in this case, we're showing the top 20 differentially methylated regions in exons, but we can do this for introns or for CPGs. And so you can see the most important genes. The next step is to annotate the differentially methylated CPGs or regions. And so we use a package called Annotator. And this allows us to annotate all the DMCs and DMRs, either in genomic regions, such as exons, introns, promoters, or in CPG island annotation. So this allows us to see in your comparison, where are the highest differences. And then to finalize, well, as with any other omic application, you can do other downstream analysis like gene ontology enrichment analysis or pathway enrichment analysis. We can do this at Diagenode as well. We can customize this type of analysis for you. This is interesting to pick a set of differentially methylated CPGs or regions between groups of samples and to look at what are, for example, the biological processes that are more involved in those. So just to go a little bit on the application of DNA methylation as a stable nucleic acid based modification that is relatively easy to handle. It can be analyzed with great reproducibility and accuracy. So it is quite widespread technology used in the field of clinical diagnosis and personalized treatment. It is the technologies that I mentioned throughout the webinar, so Methylkit, WGBS, RRBS, EPIC, they are very often used for biomarker discovery, and they are applied in fields such as disease diagnosis, disease subtype, prognosis, response to drugs. The most popular fields resorting from these applications are usually cancer biology and nutrition, for example, studying diabetes type 1. But it can also be used for other types of studies, such as transgenerational inheritance. It is used to understand better the effect of the environment. So, for example, it has been used to study the effect of pollutants or the adaptation to stress, for example, to drought in the case of plants or to parasites in the case of fish. And it is widely used to understand better plant growth and development. And last but not least, it has been used lately in the science of longevity and aging. I'm sure you've all heard about the epigenetic clock, and it's a way to estimate biological age throughout DNA methylation. So as a summary, DNA methylation is an epigenetic hallmark that is present in virus, bacteria, plants, and animals. It has, as I just showed, a wide range of applications such as cancer, neurodegenerative disease, or nutrition. We can extract information at different levels on scales such as genome-wide, reduced, or targeted. And we can also choose what resolution we want, if we want just to check methylated regions or if we want to go to the single nucleotide resolution. And what I wanted to share with this webinar is that there is a bunch of open source tools that you can use for analyzing that are suitable for DNA methylation. I just shared with you a few tools that I use for alignment, differential analysis, and as other technologies you can do downstream analysis. So I wanted to also share this webinar so that if you're getting started in the analysis, you have a basis on to how to start. I wanted to also mention that sequencing data files are generally large files in terms of a couple of gigabytes per file that require high performance computing infrastructure. Also, the bioinformatic tools and software that I've shared can sometimes be quite challenging to install as they perform best in a Unix environment. And so then you need expertise in command line software, in installation, as well as in compilation and in modifying environmental paths. The software used in these pipelines require, the ones that I shared today, you require at least 5 cores and more than 16 gigabytes of RAM. But just to let you know, so at Diagenode, we have 2 operating servers with up to 40 cores and more than 700 gigabytes of RAM. And some of the output files can be very large. So you also have to keep in mind that you're going to require storage space. So at Diagenode, we offer a panel of technologies to study DNA methylation and hydroxymethylation, such as I mentioned earlier, Methylkit, WGBS, RRBS, targeted. Our service offerings can start from sample to DNA to complete bioinformatic analysis and the generation of fully comprehensive reports. So as I'm mentioning here, end-to-end service. You will have a team of experts to help you from the experimental design to the interpretation of your results and publication of data. So with this webinar, I hope I could give you the basis to start off your DNA methylation analysis, but if at any stage of the process you feel you need help, just contact us and we will be very, very happy to assess you and to propose solutions. And just one last note so just say thank you to all of you who stayed in and that dialed in to join the webinar. I wanted to say that we know people are starting to come back to their labs in the coming days and weeks. We view ourselves as your partner and we would like to know how we can work with you to get you to reestablish in the lab quickly. So I would just like to ask you if you can take 3 minutes to answer a few questions on how we can help you. You can go to this URL, diagenode.com, EN pages/your-research. And last but not least, I also wanted to remind everyone that there is a $2,000, €2,000 grant program for DNA methylation studies. So this can be used at Diagenode either to purchase kits or to do services with us. The deadline for applying is the 15th of May. So if you're interested, you can go to this link here, the one that ends in grants, and apply. So you're still on time. And that's it. I wanted to thank you and wanted to let you know that if you're interested in services in bioinformatic analysis, you can go to our website, diagenode.com. You click on services, and at the end you have this link, Genomic Data Analysis Services, which will give you more information about our services, and you can contact us through the website. Thank you. I give my word back to Damien.
Thank you very much, Sol, for the nice presentation, nice and clear presentation. So maybe we'll go through a few questions that I will have a look at. So maybe first a general one is, can we use RRBS to see a global methylation level between samples?
I would say no. So as I explained, RRBS is going to target CPG-rich regions. So it is going to miss other CPGs. As we saw from WGBS to RRBS, you go from 20 million CPGs to 4 million. So if you're interested in global, I would say you need to do WGBS or Methylkit. I don't know if you, Damien, have another view?
No, that was my point as well. So leading me to another question about, someone is asking how many replicates would you need to do a WGBS experiment, and what about the sequencing depth?
Okay, that's a great question. So that's going to depend so much on your study. That's why we encourage you to contact us, because we personalize each project to your biological question. It's going to depend a lot on what you're studying. If you are going to be doing comparisons, we always recommend minimum 2 replicates, so duplicates. But if you can go for 3, that's that's better. This is just a statistical requirement if you want to make sure that what you're seeing is it has a statistical significance. You need more replicates. So it's going to depend a lot on what you're looking at. But if you're going to do comparisons, minimum 2, that would be my answer. And the sequencing depth, it also depends a lot, but I would say for WGBS, the minimum we recommend is 30x, if I'm not wrong.
Yeah, we always target between 30 and 50x, depending on depending on the customer.
Yeah, depending on what the objective is as well.
Yes, exactly.
Yes.
And so there was a similar question about RBS. So I don't know if you can make the similar comment for the number of replicates to do a differential analysis, methylation analysis for RBS.
But I would say it's the same for any technology. If you want to compare, you need at least two replicates, because otherwise, so you need, when you have 2 samples, you can you have a variability between them, and only with variability you can compute p-values. So you always need minimum 2 samples. And depth of sequencing for RRBS doing paired end, I think as well we are going for 30x, if I'm not wrong. Yep.
So another maybe more technical questions. Are the PCR duplicates not removed from the BAM files?
Okay, so from the top of my head, I think for WGBS, we do remove the PCR duplicates.
We do, yes.
But for RRBS, we don't remove them.
Yes, the fact is with RRBS, because it's an enzymatic digestion, you have more chance that the fragments will be the same. So you cannot distinguish between PCR duplicates and real biological fragments. So that's why for RRBS, we do not remove PCR duplicates.
Yes, that's correct.
Let me just check a few other questions that just popped up. So is there a preferred technology for the analysis of hypomethylation changes versus hypermethylation changes? What's your opinion about that?
Gosh, I wouldn't know what to say for that one. What's your opinion, Damien?
Well, I think as you are looking at methylation levels anyway with all the BS technologies, it doesn't really matter which one you are using, and it's not a question of looking at hypo or hypermethylation, but more which level of detection you want to achieve. So
yeah,
that's
correct.
will mainly drive the technology that you will use, but not really the changes that you're expecting.
No, that's what, because that's another concept that maybe I didn't go through, but hypo and hyper are always compared to something, right? So
Yes.
if you're talking about hypermethylation, it means it's more methylated than, or it has increased methylation with respect to. So you can look at it both ways. You can say it's 20% hypermethylated or 80% hypomethylated. So I agree with you. I don't think like that right now that there is a technology that would perform different if you want to look at one or the other.
Here I've got more questions related to the wet lab, but I think I can answer that as well. So regarding technical details, should the starting material be cross-linked, even if we are looking at isolated DNA with restriction enzymes?
Yeah, go ahead.
So for this technology, we are starting from DNA, so we don't need a cross-linking event to take place. So we need the DNA, not chromatin. So there is no cross-linking step to link anything to the DNA.
Yes. Maybe we have a last question.
Yes, maybe I'm just looking for a last one. So Similar question. What is the minimum sample size to be compared to states with these techniques? Have we started providing confidence of the result? So this is more related to the number of replicas that we already answered. Maybe a last one, maybe a more specific one is, can we include information from genetic variation? For example, if you have a database of known SNPs, is it possible, like if you have a CT SNP database, to include that to avoid any possible bias in the DNA methylation?
Yes, I think the answer is yes. It's very challenging, I think, especially if the SNP is CT, but we have had some projects where it was required that we removed any known SNP from the species, so that we were only focusing on real cytosine to thymine changes, and not, because it's true that you can confuse a conversion with a SNP. That's true. So yes, it is possible to deal with that.
I think that's it. If you have any more questions, feel free just to send us an email or to contact us, and we'll be happy to answer your remaining questions. And I think that's it for us. Thank you very much again, Sol, for the clear and nice presentation and for answering all of these questions.
Thank
and
you,
I
Dave.
wish you, everyone, a very nice day or evening, afternoon. See you.
Bye.